And you're not getting any extra noise off my headphones. It's New York City, there's construction even in coronavirus time.
It sounds, yeah, it sounds fine. I'm not hearing
Fine,
anything.
fine. Okay, good.
Yeah, that's good.
Maybe they'll actually get more construction done, right? Okay, I think we are up on YouTube.
Yelena, did you also get that thunderstorm last night?
I think so. I actually didn't hear it.
We didn't get much thunder, but we got a ton of rain just out of nowhere, and maybe we got the same storm.
Yeah, I think towards the morning, because when I went out of the house today, the pavement was still wet.
Wow, leaving the house. That sounds crazy. I haven't done that in a few days.
So fancy. I walked outside.
Okay. Adam, does the YouTube look good on your end?
Yeah.
Great. We are internet professionals. There's somebody in the group chat who is having trouble hearing us. Is everybody else okay on audio? Sorry, AJ, I don't know how to help you. Yelena, we'll have you go first. Is that okay?
Oh, yeah, that's fine.
Okay, great.
Would you like us to keep our cameras on? Daniel's asked us for the same, so you can see some people when you're talking.
It's up to you guys.
Don't mind.
All right. In the name of starting on time, which is sort of anathema to my personality, but let's go ahead and do that. So hi, everyone. Welcome to our, I don't even know how many installment of Cell Migration Seminar Series. 15. Wow. 15. Awesome. We are working on the schedule for the fall, and we've got a continued exciting lineup, and we'll be sending that out to everyone soon. And let's go ahead and get started. So today we have Yelena Bernadskaya. She can correct that if that's wrong. So she's a postdoc at NYU in the biology department, and her work focuses on understanding how the intersection of gene regulatory networks and cell biological processes produces large-scale behaviors during embryonic development. And so I'm seeing that in her spare time, she attempts to do science stand-up. So hopefully we'll get a little preview of that during her talk as well.
Oh God, that's a lot of pressure. I did not prepare a set. I maybe you'll think the research is funny, but yeah,
okay,
who knows?
Never mind, no jokes. Yelena, go ahead, floor's yours.
I'll try. Well, first of all, I want to say thank you for having me. This seminar series has been really great. I'm very excited that it's being extended into the fall. It's been really nice to be able to reconnect with people that I've met, as you said before, at some of the Gordon conferences, and have a little respite from some of the coronavirus things that we've been struggling with. It's very nice to have a place to go and see new science and reconnect with people. So today I'm gonna tell you guys a story about what I've been doing kind of since the coronavirus hit, and we haven't been able to do a lot of things in the lab, but fortunately we've been collaborating with computational biologists who are very productive regardless of whether they're in the lab or not. And what I'm going to tell you is the work that focuses on how adhesive forces can help promote organization, movement, and leader trailer cell state during collective migration. And this work is done in collaboration with the Gilner lab at NYU, who are computational biologists. And the animal that we work on is not one that is very commonly known, but it is a very, very cool one. So I'm going to give you a brief little introduction to our model system. And hopefully after this, it'll be everybody's favorite and everybody is going to want to work in it. So the animal that we work in, let's see if oh yeah, screen advances, very good. Here we go, sorry, I'm like rearranging my screen constantly. It's so weird. So the animal we work on is Sayona robusta, which is a simple chordate. And here you see an adult form of it. And while it doesn't necessarily look like it here, it actually contains all the trophoblastic tissues. And here's an embryo, which is the model system that I work in. And you can see here highlighted the epidermis, the notochord, endoderm, neural tissue. And it has a number of advantages. It has a very quick developmental time, so you can get swimming larvae within 12 hours. Another really nice thing that you could do is you have markers for every type of tissue. So you can mark individual tissues and watch them develop live, which is very helpful. And the other thing is that you can get really large number of embryos because we would dissect out the eggs. Here's the eggs are orange here and the sperm is white. We can dissect this out, do in vitro fertilization, and get a lot of embryos for every single experiment. Now, the major focus of the lab, let's see, slide advance, there we go. The major focus of the lab has been to understand the migration and specification of these two cells shown here. These are the cells that are born in the trunk. They migrate to the ventral epidermis, and therefore they're called the trunk ventral cells. I'm going to keep referring to them as TVCs. And these cells are cardiopharyngeal precursors, meaning that after their migration and after undergoing a number of cell divisions, they give rise to the Sayona heart. And we understand pretty well the gene regulatory networks that lead to the specification of the TVCs that allow them to migrate. And we have an idea of the later specifying networks that can differentiate pharyngeal muscle versus heart. But what I've been really focused on is the morphogenetic process of this migration that the cells undergo. And you can see it illustrated here. Here are the trunk ventral cells that are migrating away from their sister cells that stay in the tail. Here it's restarting again. It's an active migration, and the tissue that's highlighted here is, I believe, the mesenchyme. The cells migrate away. And after multiple divisions, they give rise to a beating Sayona heart, which is a little tube that beats by peristalsis, and fun fact, can beat in either direction. So what I'm going to tell you about, here's a little overview of what I'm going to talk about, is how the polarity of these migrating cells is established, how they interact with their surrounding tissue. And an interesting question is, what's the optimal migratory cell number? Because these cells tend to migrate as a pair, as bilateral pairs. The question is, why two? Why not one? Why not three? And do they have to be connected while they migrate? So before I get into that, I'm going to tell you a little bit about these cells so you can orient yourself and kind of understand how the polarity of the TVCs is established. So when they migrate, these cells migrate as bilateral pairs that are already polarized in the direction of their migration. So this is a model of the embryo. These are the cells that we're looking in. And if we zoom in on them, we can see that the pair of cells has one leading cell that generates a broad leading edge, and it has a follower cell that has a tapered retracting edge, and they're connected by a cell-cell junction that's maintained during migration. And here this junction is shown in black. And this is not only qualitative polarity, we can actually quantify multiple parameters of this. For example, we know that the leading cell, which has to generate a broad leading edge, is actually less spherical than the retracting trailer cell. The cells also migrate over epidermal tissue, and the leading cell makes devotes more of its surface to contact with the epidermis compared to the trailer. And again, we can quantify this by segmenting the cells and then measuring the surface contact that's devoted to interaction with the epidermis. So the leader makes more surface contact. And finally, there's a signaling difference between these two cells. And it's most apparent when we look at BMP signaling by measuring the amount of phosphorylated SMAD in leader versus trailer. And what you can see here is that pSMAD levels are higher in the leading cell compared to the trailer cell. So this leads us to think of these two cells as, although they're equivalent in fate, they're both TVCs, they are adopting very distinct cell states, that of a leader and a trailer. And the migration of these cells is highly stereotypical. So here is a live movie where we've marked the nuclei of the migrating cells. This is the leader, this is the trailer. And we could see a number of things about this movement. It's highly constrained. The cells don't wander around too much. They stay within a very kind of limited space where they're able to move. And they also maintain their position during migration. Whichever cell is specified as the leader, it stays in that relative position. They never switch places. And this is a very stereotypical migration. We could observe this in multiple embryos, even though these are non-isogenic lines. So to kind of study how these parameters are established, it would be very difficult to modulate every single parameter biologically. And what we've done is we've teamed up with a computational biologist in the Mogilner lab in order to model cell migration and be able to ask questions about cell behavior when we modulate either protrusive forces, retractive forces, or adhesive forces. And this will allow us to generate predictions about cell behavior that we might be able to test in vivo. I don't know why there's a red line there. Okay. So I'm working with a postdoc in the Mogilner lab, Haisen Yu, who's very talented and she's very good at 3D modeling of the cell migration. And the first thing that she did is generate an in silico model of TVC movement. And she did this by varying the amount of either protrusive forces or retractive forces in the leader and trailer until she was able to generate a computer model that evolved this similar cell shape compared to cells we observed biologically, with a broad leading edge here in the leader and a tapered trailer, and also this cell-cell junction that's curved in the direction of migration. And once she settled on these conditions, she was able to start varying some of these parameters and see how the cells behaved in the model. And one of the first things that she did was when she finally landed on something that allowed the cells to behave very similar to biological cells that we observe, and this is a movie of one of these cells moving, she noted that the leading cell needs to suppress a retractive activity, and the trailer cell needs to suppress protrusive activity to maintain this orientation of a leader following leader migrating one direction with a trailer following it. And this turns out to be something that we can test. So in her model, if the trailer failed to suppress protrusive activity, what she saw is that the cells were not able to organize themselves linearly. And what you got is, instead of a leader and a follower, is two cells migrating in parallel to each other. So we are able to test this by increasing protrusive activity in our migrating cells by overexpressing constitutively active ρDF, which induces protrusion formation. And true to the model, what we see is that compared to normal cells, when the leader and the trailer are aligned linearly, if we have an upregulation of protrusive activity, the cells travel in parallel to each other. The other thing that she varied was the ability of cells to adhere to their substrate, because the cells don't migrate just over nothing. They're actually migrating over an epidermis and make contact with the extracellular matrix. And what Haisen saw in the model is that if one cell had less adhesive ability than the other, you again lost this linear organization. So here in green is the epidermis that the cells are migrating over. This is a presumptive leader and a presumptive trailer. If one of these cells is not adhering to the substrate, they detach and they can start to tumble. They start to change position, which is not something that we see in normal migrating TVCs. Again, we can verify this biologically because we can disrupt adhesion by expressing a dominant negative form of this receptor, collagen receptor called DDR. So again, like I showed you before, normally the two migrating cells both make contact with the underlying epidermis. And so here is leader and a trailer. And if you don't have the ability to adhere, you start to peel off the substrate, which is very similar to what we see in this model here. Now, this has a very drastic effect on the migration, because as I showed you before, the cells usually migrate in a directional fashion, keeping their relative positions of leader trailer. Now, if you disrupt adhesion, as predicted by the model here, what you see is that the cells can no longer maintain their leader trailer polarity, and they're going to start to tumble right here. They're going to switch positions. Yeah, I'm going to be able to play it again right here. So, and you can see them roll over right there. So this leads us to the conclusion that adhesive activity is required to not only to maintain the leader trailer states, and if you disrupt either protrusive activity or the adhesive activity, what you end up with is two cells that are basically equivalent. There's no longer a leader and trailer. The cells are now equivalent in terms of their sphericity. This is verified biologically. They're equivalent in terms of their signaling. The BMP signaling in the leader is reduced to be the same as that in the trailer. And this prevents the cells from migrating as efficiently as they would normally. Their total displacement at the end of migration is significantly reduced compared to wild type. So what does it mean when these cells are becoming equivalent in terms of migration mode? Well, if you're two cells and you start out in a certain orientation and you need to migrate and you need to have a leader in the front and a trailer in the back, you can arrange yourself in a number of different positions, right? And you can use different migration modes. So what Hysen did is she developed 3 different modes of migration that she tested for their ability to organize, for the cells to organize themselves linearly. The first one, called independent or same mode, which would mean that the leader and trailer are both just moving forward, right? They're not following each other. The direction of movement is shown here. There's a faster, slower mode that she tested, which says that the leading cell is moving slightly faster than the trailer. And there's a leader trailer mode, which means that the leader begins to move and then the trailer orients itself towards the leader. So she tested which one of these migration modes is able to produce the biologically relevant linear orientation of leader and trailer. And she reports this as cosine of this angle theta that's made between the trailer and leader connected by their centroids compared with the direction of their movement. And these cells are, they maintain a cell junction between them as they move within her model. And what she sees is that if your cells are moving independently, if they're not oriented towards each other, they can never achieve this linear organization of leader and a follower. They're always going to move in parallel. Now with the faster, slower mode, they eventually achieve this organization, but the best is to have the leader actually follow, the trailer actually orient itself towards the leader, which is moving in a specific direction. And this allows the cells to reach this organization much faster. And this is an interesting observation because this is what we're kind of seeing when we increase protrusive activity in both cells. The cells are taking on this parallel migration mode, and there's no longer a difference between leader and trailer. So in the establishment of TVC polarity, what we've learned from our modeling and biological assays is that you require suppression of protrusive activity in the trailer in order for the cells to adopt leader trailer states, and also requires adhesion to the underlying substrate to maintain the TVC polarity during migration. And you also need to polarize the trailer towards the leading cell to achieve most efficient migration. Now again, these cells are not really moving in a vacuum. They actually migrate between the epidermis, I'm showing here, and an overlying tissue, the endoderm, that they have to displace during migration. So here is an image of the endoderm in green. Here are the TVCs in magenta. And if you just zoom in on the endoderm, they say epidermis, this is the endoderm, just in case. If you zoom in on the endoderm, you can see that the TVCs actually carve out a little pocket for them to migrate through. So they're displacing this tissue. So Heisen modeled this behavior as well. So here is the epidermis that the cells are moving on. Here's the leader, here's the trailer, and here's the overlying endoderm. So when the cells encounter it, they're basically encountering physical resistance to their movement forward. And within the model, Heisen was able to assay different stiffnesses, and these are relative numbers here. This is a coefficient of conservation of volume, and their interaction of the endoderm with the migrating cells. So if the endoderm is too stiff, and this is here with lambda 0.5, what happens is the cells run into it, the leader cell contacts it, is unable to displace it, and then the trailer comes in, and this is shown here, and tries to pass it because it's still capable of migration. And this ends up jamming the cells parallel to each other, and they're not able to move. However, if the endoderm is soft enough, the leading cell will burrow under it, right? And the trailer eventually will follow. But what this does is it creates a pause in the migration. So here is the basically instantaneous velocity of the cells. So it's position over time. And here is the leader cell moving forward. Here's where it encounters the endoderm. And we could look at the slope of the line and look at how, and get an idea of how fast it's moving. So once it encounters the endoderm, it begins to slow down. Once it's fully migrated under the endoderm, it speeds back up again. And this is also true for the trailer, unless the endoderm is too stiff, and to this blue line here, and in that case, both cells stayed the same. But what this predicted is this pausing behavior when the leader first encounters the endoderm. And this was very interesting because years ago, when we just started assaying the migration of TVCs, when we averaged the instantaneous velocity of 8 embryos, right? So here is leader in black and trailer in red. What we saw is there's a point in this migration when the leader slows down and the trailer speeds up. Now, we didn't know what the reason for this was at the time, but now we are hypothesizing that this is the point where the TVCs encounter the endoderm prior to their full migration under it. And the leader is slowing down because it's encountering this resistance, which gives the trailer time to speed up until it catches up. So the interaction with the surrounding tissues really helps maintain polarization of migrating cells depending on how stiff the surrounding tissues are, because if they're too stiff, you can end up with different orientation of cells during movement. And there's requirement to displace tissues during migration, which can also inform the ability of the cells to move. Now, everything I've been showing you is two cells moving. But what is the actual migratory cell number? Why do these cells tend to move as a connected pair rather than one individual or two individual cells? So again, going back to this observation that they move together and they need to displace a certain amount of endoderm, we could think about it this way. If these cells are moving in this direction, they're encountering resistance from the medium here. So this is where the endoderm is, and they have to push against it. Now, if two cells are moving together, they only meet this resistance once. If the two cells are moving separately, each one has to overcome the resistance from the medium independently. So using this model, Heisen tried to predict what would happen if a single cell were to migrate. And what she saw is that computationally, a single cell was less efficient in migration, both in terms of its ability to move fast enough. So a single cell in her models is slower than two cells, as well as its ability to move persistently, to move forward in the directional state. So if a single cell is moving as is shown here, it is a lot less persistence. In fact, its persistence decreases over time compared to either of the three modes that she tested for two cells. Now, we've known for a long time that the TVCs, the pair of cells, they don't need to be a pair. They're both competent for migration. And we can induce this by blocking signaling from one of the cells, and you get migration just of a single TVC. But as the model predicts, this cell seems to be much less competent, much less efficient at migrating. Because when we compare its total displacement, we find that in wild type, the cells are displaced by about 50 microns in total. A single migrating cell is displaced only by about 40 microns, so they tend to under-migrate. So this may be because two cells are better at buffering signaling cues compared to a single cell. And this tells us that an optimal migratory cell number when the cells are connected is probably 2+. And this leaves us with some really fun experiments that we're hoping to do sometime soon, such as manipulation of stiffness of the surrounding tissues and seeing how it affects ability of TVCs to migrate and evolution of their morphology, and analysis of larger migratory clusters, because not only can we induce single cell migration, we can actually convert the sister cells of the moving TVCs to a TVC fate and get clusters of 3 or 4 cells moving together. And we can assay how polarity is established and maintained in these larger clusters. So that is the end of my talk. I'd like to thank our lab, so my PI, Lionel Christian, all our lab members, and the Mogilner lab, and particularly Hai-Sen Yu, who's done all the computational things for this. We're writing this paper up right now. If you have questions about the modeling aspect, I will do my best to answer them, but I am just a simple cell biologist. And I don't know if Hai-Sen was able to join us today, but I will do my best, and thank you for your attention. I'll try to take questions.
Great, thank you so much. We're actually running short on time, so we're going to do a couple quick questions from the chat. So first from Claire Waterman, she's asking if the cells are migrating on the surface of the tissue or between other cells.
So they're migrating in between two tissues. They're making adhesions with the extracellular matrix that overlies the epidermis, and the endoderm is on top of them. So they're sandwiched between two tissues as they migrate.
Great, and then we'll just do one more, and maybe the third one you can answer in the chat. Gareth
Sure.
Jones asks, if you laser ablate the leader of the pair, what happens next?
The short answer is we don't know. We have not done an experiment where we killed one cell. Well, it depends when you do this, right? Because if you don't specify one of the cells as a TVC, the other one will migrate. It doesn't matter whether it's in the presumptive leader or trailer position. Now, ablating one cell in the middle of migration, that would be interesting to see if the other cell keeps going, or let's say if the trailer in that situation is now able to crawl over the other one and continue migrating. But initially, if you start just from the beginning, they're both equally competent for migration.
Okay, great.
Thank you very much.
Thanks so much. Sorry, we're running out of time here.
No worries.
If you want to stop
sharing.
Great, and Daniel can come back.
Yeah. So our second speaker is is Daniel Cohen. Daniel is an assistant professor of mechanical and aerospace engineering at Princeton University. His new research group has just entered its third year, where he and his team are studying a variety of aspects of collective cell behavior, with a particular emphasis on knowledge and approaches that can help us better engineer and control the behaviors by means of biomaterials to bioelectrical interfaces. He also studies water bears, which are another form of cellular collectives. And please feel free to turn your cameras on so Daniel has a bit of an audience to talk to. Take it away, Daniel. Thank you.
Yeah, hopefully there will be at least a few laughs, so it'll be good to know if the jokes are landing. Thanks a lot to the organizers for inviting me. This was definitely a Zoom meeting that I was actually looking forward to compared to most of them, so that's exciting. So I appreciate the interest in the venue as well. A little bit of basic background to this. This will be a relatively high-level engineering talk, because I think that something that's really exciting about at least collective migration and directed migration is that there's an engineering component to that for practical applications that my lab really tries to explore. And I'm going to try to sell you on that connection. But also, I'd really appreciate feedback on how that message goes over, questions you think we could be asking, et cetera. And so something that I'll just start with is to note that when I started grad school, I swore that I was never going to work with cell culture again. I'd done a little bit in undergrad as a mechanical engineer for tissue engineering and decided I was done. And now my lab is 2 years old and that's what we do. And the experiment that really pushed me over the edge and sort of brought me back to the light is shown here. It was a workshop at Woodsville.
Sorry, Daniel, you're not sharing your screen yet. We thought you were just giving an introduction.
All right, okay. Let me try again. Oh, Zoom etiquette.
Yeah, there you go. Thanks.
Okay. Yeah, this is the experiment that changed my perception of a lot of things. So most of you have seen things like this before. It's just basic epithelial wound healing, but I'd never seen time lapse. I'd never seen collective cell behavior. I scratched this MDCK epithelium here. The blue is nuclei, red is E-cadherin, and it started healing. And it was on a time lapse system, but I stayed up all night watching it because I was just really blown away. And when you're an engineer and you see something like this, it's really humbling because what you're looking at is a system where you have, say, tens of thousands of agents that sort of self-organize to solve a pretty sophisticated problem. The tissue doesn't really care which cell winds up where, but it cares about the state. So you go from a state of being injured or having an open boundary to a state of being healed or closed. And it does that every time, just in slightly different ways. And everything's fine. That's how a community kind of solves problems. And so I started thinking about this more as a sort of communal dynamics exercise. And I went back to some old ideas from the 1800s from Rudolf Virchow, who's the founder of cellular pathology. He challenged Otto von Bismarck to a duel with poisoned sausages. And he also came up with this concept of the Zellenstadt, which is the phrase meaning the cell state. And so he did have some political motivations for this, but the concept is apropos. When you think about a multicellular collective, an organ or an organism, they have aspects of hierarchical organization. You could almost think of it as a community or a nation state in his view. You've got tax collectors, you've got waste management, macrophages, you've got military force in the immune system, you've got transportation in the blood vessel network, structural support in soft tissues and bones, right? You can carry the analogy as far as you want, but you have this community that works together to produce a certain set of functions. And if we think about manipulating these behaviors like collective cell migration as a sort of communal problem-solving system that we can bias, there are some interesting options that become available to us. So what my lab tries to do is to look at collective cell behaviors and try to both better understand them and then come up with new ways to take that understanding to heal or build tissues in better ways. And so today we'll focus on these two areas, the understanding and perturbing of sort of basic collective cell behaviors as a concept, and then some specific focus for most of the talk on one way that we can go about actually hurting these systems with some real-time control of cell migration. So just so we're on the same page, just a little bit of basic biophysics. This is reminiscent of what Benoit talked about not that long ago. So this is, I apologize, the movie has frozen, but there are really just three things you need for some sort of collective behavior, regardless of whether it's birds, fish, or cells. The first is that you have local interactions that are going to produce global order. And in the case of the wound healing movie here, it's depending on how strongly the cells are attached to each other. So a loose collective versus a tight collective will give you different behaviors, and those propagate out at different distances and give you the global dynamics. Second, the system is greater than the sum of its parts. So if you were to take a cell out of this population or take a bird out of a flock and understand it down to the molecular level, knew what every neural activation pattern in that bird meant, it wouldn't actually tell you what that bird's going to do or that cell's going to do in a group. So you really need that systems level approach to think about these. And the last thing is that nobody tells these what to do. They have almost a teleological set of collective behaviors that they will simply execute. And that's super important from an engineering standpoint, because if we can identify what those impulses or behaviors are, we might be able to benefit from them. And instead of having to reinvent the wheel, we might be able to ride on top of what's already there. So how do we engineer collective behaviors? What does that mean? What's some high-level thinking around it, and what are some examples for that? And the concept that I just emphasized was that you kind of synergize with what's there. And this becomes useful in a number of applications. If you try to think about wound healing, it's already a collective process. Can we accelerate that collectivity to make healing occur faster, for instance? That's a sort of simple example of why we care about this. So how do you tap into these things? A case study that I'll start with, because it's really pretty and it's new data from the lab and I think it's worth taking a look at, is to just start by understanding some basic behaviors in a tissue and build that up to something useful. So this was one of the first studies in the lab. It took a while to come out, but hopefully it'll be out pretty soon. Got a lot of support from these great people here, including Ricardo Lert, who will probably be speaking in not that long here. And so we started by just asking, what are the dynamics of large epithelia? This is an MDCK kidney epithelium, and it's growing over about 2 days here. And you see huge vorticity forming in the middle. That's something that hadn't been observed before. You see very smooth boundary growth. We did a lot of biophysical quantification of this system, and we know a lot about how and why it does what it does. And you can see that on the archive. Hopefully the paper will be out soon. But what we do with that then is we say, okay, if we understand how these things grow, we understand the relation between the inside and the outside, or the fact that the outside is largely decoupled, then we can just look at two collectives and look at how those collectives interact. So this is a video of two differently shaped epithelia, but everything else is the same: density, cell type, color is just a membrane dye, colliding with each other and producing this stereotyped boundary that we can actually model now. And we've developed a simulation approach that allows us to predict the sort of steady state of a variety of different epithelial configurations. And using that, we can build a little simulator to kind of design tissue. So this is a simulation of what would happen if you had tissues like this. And that's like tissue CAD design, if any of you have worked with engineering software. And we can put that all together now to produce something like this, which is a tissue mosaic, where we pattern the tissues and then let them grow over a couple of days. And we could predict almost exactly what they grow into. So this beautiful combination of green diamonds in the purple matrix is something that's now predictable. So we can grow tissue mosaics simply by knowing a little bit about the collective interactions that drive single tissue growth, and then what happens collectively when multiple tissues interact. And we're looking at tissue engineering applications and some biophysical studies and trying to look at sort of importance for compartmentalization in tissue development. But as a simple engineering case study, maybe that gets the point across that you don't need to know everything that's going on inside in terms of signaling and the subcellular dynamics to get something useful out that's predictable and hopefully practical. Okay, but we like to play around, we like new tools, and we like to really push the limits of what we can and can't do. So can we interactively steer migration is the next question. And this is one that's occupied me for a number of years, which is what level of control can we get over cell migration? How would we do it? And what does it let us accomplish? And to give an example of that, this is our oldest swarm technology, I would argue. This is herding of sheep. It's about 7,500 years old in terms of domesticated animal herding. We still don't know how it works. Papers are written every year trying to describe it, but look at just the beautiful behaviors. You've got contraction, expansion as the sheep go through that gate. You've got a jamming transition. You've got order maps and orientations. And the interesting thing is the shepherds don't read that literature. They don't know any of the technical details about how this works. Certainly the dogs don't, but they're able to get this exquisite control by just looking at the system, perturbing it iteratively and adjusting until something works. And we've gotten really good at this as a species. So again, if you understand enough about the collective perturbation response of a system, you can make that system do some pretty wild things. And what we'd like to try to do is to build that into a collective behavior or tissue model. And so again, if we think about herding in cells and tissues, there are a number of things that's useful for. So tissue sculpting is that concept of taking a blob and inducing morphogenetic changes that involve collective migration. So controlled branching or vascular network formation or changes in shape or size. Accelerated healing we've already talked about, but there's a nuance to that. Maybe you could double the rate of healing by accelerating cell migration, but that might cause greater scarring. You could also think about modulating scar formation by adjusting contraction as the wound is closing. those are all things that involve coordination. And so now we need to start thinking about sort of performance metrics and what it means to actually control something and what's a desirable result. Simple example is you take this to a lab setting and you just want to say study front rear polarity in migrating cells. Well, what if you could make a cell go back and forth and back and forth and back and forth? It's a pretty useful tool for generating a lot of data quickly, we hope. So those are the kinds of motivations that we think about for this. And now we need to think about how we'd actually get there. So we have a system like this as our goal. We want to be like the sheepdogs. And we're stuck with something like this. And how do you go from that thing on the right to that thing on the left? And how do you do it with a couple of constraints? The first being, we want this to be interactive. You can get some amazingly sophisticated control using micropattern substrates, like Benoit showed with cells going around and around like a traffic system. But we would like something that you can actually program and adjust in real time. It needs to be something that's not going to kill the cells with prolonged use. And lastly, it should have broad applicability. There's very powerful stuff you can do by genetically modifying cells, introducing optogenetic targets, things like that. But we'd like to see what happens if we just start with the behaviors the cells already have. How much biasing or interaction and input do we have to those endogenous processes already? So first thing is choosing the kind of sheepdog you're going to use. And chemical cues we know are very powerful. You've had a number of talks on chemotaxis in this seminar series. It's a really neat phenomenon. It has a couple of issues here though. The first is that it isn't all that widespread. I was surprised at how difficult it was to find good model systems for collective chemotaxis across a large number of species, because it's ligand dependent, there are a lot of subtleties, and it's very difficult to program spatiotemporally. So as cool as collective chemotaxis is, it's a little bit difficult to work with as an engineer. Mechanical cues are extremely powerful. There have been a number of seminars on those here as well, how they shape morphogenesis, collective behaviors, substrate stiffness interactions. We can control a lot of behaviors, but it's not something we could dynamically adjust. Again, getting that spatiotemporal control over the mechanics of the system is a little bit difficult. And so that brings to mind maybe electrical cues, because we're just going through the different schools in the engineering department. And when we think about programming, it kind of makes sense. But when we think about bioelectricity, because that's what we're talking about, we get some problems. This is an image that typically comes to mind, or something like this when we talk about non-neural bioelectricity. When I go up to people on the street and ask for their opinion, which I do, which is a little bit weird maybe, I'll sometimes get the very sad response where people say, oh, bioelectricity, it's like the electric chair, right? There's an image problem with this field. And it's really sad because it's a fascinating field with an extremely long history. This was discovered well before chemotaxis. And I'll talk you through a little bit about that troubled backstory so you understand some of the challenges in the field, but also some of the fundamental discoveries that we can tap into for the work you're going to be seeing. So a lot of the problem with bioelectricity began with the fact that it first manifested as twitching frog legs from the Galvani and Volta dispute, which most people have heard about, which then led to these 1806 corpse galvanism experiments where Galvani's nephew, Giovanni Aldini, actually electrified freshly killed or freshly executed criminals to cause the corpses to sit up and wave their arms and blink. That really made an impression on people, as you would expect. And the problem is that it wasn't necessarily a good impression. It shows you that bioelectricity is doing something, and maybe like people got all the wrong impressions. And Frankenstein was published about 10 years after this. And even though electricity is mentioned nowhere in Frankenstein, Mary Shelley answered when interviewed about what might have allowed the monster to come to life that, oh, you know, galvanism has shown promise here. And galvanism is that concept of applying an electric stimulus to something. So it was in the air, and it wasn't too long before this sort of thing started to happen. This is early quack medicine with bioelectric components. This is a corset using bimetallic plates to improve posture and bust size. There were male equivalents, the virility belts that used electrochemical plates to improve or enhance male function, kind of like those spam emails you get. We haven't really gotten that far from there. So you have this problem where there's this long history now of negative associations with bioelectricity that have probably chilled research to some extent because it's kind of hard to see whether it's a pseudoscience or not. And what's really unfortunate is there's some very serious cell physiology and biology hiding behind this. And this is one of the big discoveries that I would point to, which is Émile Dubois-Reymond, who wrote the first electrophysiology textbook, shown on the left here, performed an experiment inspired by Carlo Matteucci. And it was one where you cut your skin and you have a wound, say, in your arm, and you can measure a current in the injury site, an electrochemical current at your injury. And it's very real. These are modern data reconstructing this in rat corneal wounds and human skin wounds. These fields occur immediately upon injury. And Dubois-Reymond noted it, called it the wound current, wrote it up, and it was largely forgotten. He's a very interesting character. He may not have been the best spokesperson. He was famous for performing experiments shirtless because he thought he was a better physical specimen than everyone else in the lab. You know, we need a concerted community effort to change the image problem here. But the wound current was floating around then in the literature for a while, and it took a long time before we had enough sort of electrochemical and anatomical understanding of what might be going on to pin it down to something. So skin by default has about a 50 millivolt transepidermal potential across it. And that's maintained actively by ion pumping and the impedance of the cells within skin itself. So healthy skin maintains this potential. When you get an injury, you break that potential locally. The problem is the cells at the edge of the injury are still actively maintaining something. So now you have a tension between preserved potential out here and lack of potential in the wound itself, which tends to make the centers of wounds negative and the peripheries positive. And this produces the wound current. And these have voltages on the order of, say, 1 to 10 volts at a variety of different currents. It's measured across all sorts of species, zebrafish, frogs, mammalian cells. Dictys respond to these things. And what's important is that we think that this affects receptor distribution in the membrane. So a DC electrochemical field can't enter the cytoplasm directly because the cell membrane impedance is too high. But what it can do is it can cause electrophoresis or electroosmotic flow effects on receptors in the membrane. And it might be able to bias their distribution in such a way that you alter front-rear polarity, for instance, in cells. And it's known that cells can actually migrate in the direction of electric fields, typically towards the negative terminal. And that's called electrotaxis or galvanotaxis. It's the directed electric migration phenotype. And just to emphasize what the simple model says, if you turn a current on, these arbitrary receptors that have yet to be fully identified might migrate to one side of the cell and produce a cue. Now, work over the last 30 years has done a really good job pinning down a lot of the pathways involved with this. So PI3 kinase, PTEN, PIPS2 and 3 play very important roles, as you might expect from work in chemotaxis. These cues can activate EGFR in a ligand-independent fashion. Ion channels modulate this effect. Aquaporin flow has some effects on this. Even pH and viscosity of the media can determine the direction a cell moves in an electric field. This is a very well-studied phenomenon for something that not that many people have actually heard about. And most of this work's been done with single cells. And while there are a lot of deep mechanistic questions about how this functions, we bypassed some of those to just say, can we apply this at a collective level? Because I was looking for something that would allow us to herd cells collectively. And so my work started by bridging this to whole tissue systems. So if we have Tricky the tissue triceratops here, can we make her move? Not yet, but we're getting there. And I'll introduce a little bit about how these systems work, show some of the basic data on this, and then we'll go into some of the newer data. But what I want to emphasize is that the technology behind this is extremely simple. It's very easy to build fast, cheaply, and even reproducibly using some of our microfluidic methods. And we're really happy to work with anybody here who's interested in this and wants to just reach out, because you can make this work quite well and you can really do some fun biology with it. So almost all electrotaxic systems look like this. You have a current source that provides, this could be a gel box power supply even, that provides current that flows through your anode into a salt reservoir, through a salt bridge, into a microfluidic device where your cells are cultured, out the other side and through your cathode. You're using chloridized silver wire, very easy to buy or make. And the bridges here protect the cells from any electrochemical breakdown. So you don't even have to really optimize your system much as long as you've got an agar bridge here. The microfluidic device is necessary because it concentrates the electric field in a very small zone, which means that you know exactly where your field drop is occurring and you can tune the field strength that the cells experience. So say 1 to 10 volts per centimeter. and you put your objective underneath it, and you're good to go with the basic system. If you're going to study this for collective behavior, you also need some simple analytical techniques. So we use the same things used in collective cell biology normally. The first one is particle image velocimetry, or some version of image correlation, where you take a time lapse and you just correlate zones of an image to the next time point, and you look at displacement maps. And you can build a vector field that tells you roughly what the flow patterns in that tissue are like. And you can then use that vector field to generate a lot of data. So one thing is a visualization, which is a line integral convolution, which kind of shows you the flow pattern in the tissue, with color being speed and texture kind of being where a particle would be carried to if you were to release it in this. So you have the nice tools to really start to think about how these systems are responding, because you need something quantitative to tell you if it's listening to you or not when you give the tissue a command. The last thing then is we use something called the order parameter, or the directionality order parameter, which just looks at, if I tell the cells to all move to the right, to what extent are they actually all moving to the right? And you're looking at the vectors in the vector field or their trajectory directions. You average it over the system, and a perfectly responding system has an order parameter of 1. A system that says, nope, we're going the opposite direction is -1, and one that just ignores you is 0. So those are all the basic tools we need. So a simple example of this in practice then is an MDCK epithelium here. We're going to tell it to move to the right, and it moves to the right. That was pretty gratifying when we first saw that. That's just what it does. There's a lot of little fluctuations in there, but it's clearly following the command. If we look at the flow pattern in that system, you can see as the field kicks in, the speed starts to go up and those streak lines start to become much more parallel. If we quantify this and we look at the order parameter, you can see that before the field turns on, there's some fluctuations, probably due to stage motion and just fluctuations in the tissue itself, because it's a highly collective tissue. But then everything snaps into sharp control when you start following the order to the right, while vertical fluctuations drop to sort of 0 order. So it's clearly working. We have a way to measure the strength and accuracy of our control. Sounds good. We then tried collective U-turns just to see what happens. And you can, in fact, make a tissue bounce back and forth. There's a lot of stuff going on here, and we only touched the surface of it in the original study. So how the tissue makes the U-turn is actually locally coordinated. You don't have a global U-turn. Little patches of cells in the tissue will turn around, and this isn't a simple repolarization. They actually trace out U's. And not every cluster makes the U-turn the same way. So in this case, they could turn up or they could turn down as they go around. Somehow it coordinates all of that without us needing to tell it anything. If you ask even 20 humans to do this while holding hands or walking in close proximity, you're going to get all sorts of traffic jams. So again, we can make a really complex system do something really complex with a very simple input by tapping into what the system already does. And you can go through the original paper there. There's some neat quirks and little follow-ups to some of the details I'm talking about. But one thing I do want to foreshadow here is it's not all roses. There are problems with this, and they're very rarely talked about. But only a couple of recent studies, one from Alex Mogilner's group, has actually started looking at boundary effects in these systems. And so when you have these large tissues, often the edges have difficulty, especially in strongly junctioned epithelia. And what we noticed is that a large MDCK epithelium without a field on it has this pattern of speed and sort of flow. And when you turn the field on, you get a huge amount of electrotaxis in the middle, but the edges remain basically stationary. That's not really a good thing if you're trying to argue that you're going to move a tissue around for practical purposes. This might be great if you want to study some aspects of junctional or cytoskeletal biology, but if you actually want to move something around, this may be a problem. And it's something that we're going to talk more about later. But the next thing we'll move on to is to probe more precisely what level of control we have, and a little bit more about the signaling mechanisms that might be active here at a sort of physical level. So what we then asked was, is collective migration something that's particularly plastic? Like once a tissue starts going, can you actually bias its motion on command cleanly? How much actual control do you have? And so we built a new device, partially because we wanted to use a new acronym, and partially because we needed to have multiple electrodes. So Sheepdog is probably, well, it's my favorite acronym right now, but it's a very nice, clean electrotaxis system that uses 4 electrodes, an x-axis and a y-axis, and 4 sampling ports, inline perfusion to handle any risk of temperature changes, get rid of metabolic waste, bring in freshly oxygenated media. It clamps into an existing Petri dish. You can put a glass plate on the bottom of it for high resolution imaging. It's a nice system. And this is just a cross section of it. Everything's built by basic laser cutter or little razor writers on the benchtop. And you put your tissues in the middle. You have integrated bridges and reservoirs, and everything fits on the scope quite nicely. Again, this was just published about a month and a half ago, and you can see the methodology more there. If anybody wants to build one, we're happy to help. We've published all the design files as well. So what we did here was not just use a new device, we used a different tissue model. We wanted something that was more organotypic and physiological, and also closer related to skin, since that seems like a good application space for electrotaxis. So we started with primary mouse keratinocytes, and we're working in collaboration with Danelle Davenport's lab here to have access to these cells and some expertise in working with them, and that's been really great. And this is a population from a monolayer of primary mouse keratinocytes cultured in their basal maintenance media, and we're going to tell them to go through a controlled right turn maneuver. And so that is a tissue that is responding on command. It goes right and then it goes up, and you can watch the phase lag. The white vector here is the field direction. You can see how the tissue chases it, and there's a little bit of lag there. We've quantified that. You can design a controller that takes all of that into account if you want. But the basic fact is this works. The cells are reprogrammable in real time. And the anatomy of what that looks like is, if you look at the trajectory overlay, which we call the hairball plot, this is the first stage moving to the right, and this is moving up. This is an order parameter response showing you how the system polarizes and moves to the right very well originally. And then when you change directions, there's a little bit of lag. It still moves to the right a bit, and it's a little slower to come back up. So that's where you get this swinging out behavior here. But you would expect that in any sort of collective system where there's some interactions. You also see the speed goes up dramatically, which is something that hasn't really been fully explored or explained. And we know that it's not temperature related in our device. So something interesting is going on to make these things move so much faster. But that's what happens when you tell these things to go through a particular maneuver. And that works at the macro scale, which is important compared to the MDCK data. This is what we see here. This whole tissue will actually take off and move on command. If we ran it just one direction for even longer, you'll see how it moves in the Petri dish by eye, just by watching it. And that's something we were very excited about. There's a lot of different things going on here. We'll take a look at a few of them. But one thing that we wanted to ask was, that was a field that goes right and then up serially. And so we said, what if we change the field very quickly? Because we only have 4 electrodes, but we want to program an arbitrary maneuver. So if we change the field between right and up very, very fast, maybe we can do it faster than whatever the transduction pathway is, such that the cell experiences a field that's actually just going at an angle. So instead of feeling this, it goes so fast that it gets the average. So we switched every 30 seconds and otherwise just used a standard 2 volt per centimeter field. And this is a keratinocyte monolayer moving at 45 degrees now. And one thing we notice is that in addition to moving at 45 degrees, the cells tend to align long axis perpendicular to the direction of motion. So if the direction of motion is in peach here, blue is the long axis of the cell. And that's something that's been noticed since the 80s in single electrotaxing cells. So within this monolayer, these weakly coupled cells are able to reorganize themselves and orient and move in the field direction. And that also means that they're time averaging this because we don't see much of a wobble at 30 seconds. So we don't see them fluctuating. They're relatively stable. So this is a tool that we can now do frequency sweeps with to try to understand a little bit more about the time scale of the signaling process. But from a practical perspective, we can now put any direction into this tissue. So what we then said was, let's make the cells move in a circle to see if this is actually robust. So we adjusted the field every couple minutes through this sequence of angles that we then stimulated a tissue with. And this is the tissue responding to that on command, going through a circular motion. And if you look at single cells, which is something we're trying to do at much higher magnification now, this is a phase image of a single cell spinning in one of those fields. And so you can see the lamellipodia very faintly is turning a little bit before, and the trailing edge is spinning around. This is just an overlay capturing the trailing edge positions. So this thing is really spinning, and we hope that we'll actually be able to understand some of the cytoskeletal biology by using something like this to watch what's reconfiguring when. We've also modified Sheepdog recently to test in vitro wound healing to see whether we can actually do some of this in an in vitro model at least, and see if we can make something close faster. So these are primary keratinocytes again, just as you've seen before. This is their standard. Well, this is PowerPoint, not being happy, but you can kind of see they moved. You'll just have to take my word for that there. Hopefully this one will actually play. And these are the tissues actually coming together. And Zoom is blocking my window, so I can't seem to adjust that. But hopefully you can see that the whole tissues are actually coming in and closing and mixing at the boundary there. And if we do some very quick quantifications, because this is very new data, we just do chymographs. The system does not close in a control case, and it closes and moves much faster in the stimulated case. And now we're looking at what happens at that interface in the middle, and if there's mixing and how the cells respond kind of at that singularity where the converging field is. We've also just recently started to get live cytoskeletal imaging working, and we're switching to higher magnification as soon as we can solve some microscope focal issues. But this is 20x GFP actin in primary keratinocytes. And it's basically a couple frames and then the field turns on, and you can see those cells really start to take off all towards the right. You can see them aligning long axis perpendicular to the field. If we watch this cell in the middle here, we can see its lamellipodium reconfigured to the right. So we think if we start looking at microtubules, cytoskeleton, nuclear positioning, we're going to see a lot more about what's actually happening when, and that might give us much more of a sort of physical mechanistic basis to how electrotaxis is reprogramming cells. So all of that's really been a lot of fun to work with. But what I want to end with is some of the limitations of this, because this is where we start to bump into the natural collective behaviors of a system that might be causing some problems. And this really was illuminating for me, especially for thinking about what a collective behavior means, because this isn't just a series of motions that a tissue goes through. It totally changes the mechanics and behavior of that system in ways that can make it harder to work with. So what I'm asking here is, how do those endogenous collective behaviors affect our ability to control it? And we're trying to say to a population that already has existing behaviors built in, hey, you can go do this thing, kind of like this sheep, but it may not be able to do that, or it may have different signaling incentives. So one thing we looked at is the difference in the leading edge and trailing edge and bulk dynamics in MDCKs and keratinocytes. So MDCK is very strongly junctioned, strong cell-cell adhesion. Keratinocytes in basal media, at least, relatively weak cell-cell adhesion. If you look at the leading, bulk, and trailing edge dynamics of monolayers, MDCK populations have huge speed in the bulk, like you saw in the heat map, but the leading and trailing edges have very little forward motion. Keratinocytes, by contrast, are relatively stable, just a little bit faster at the leading edge, but the whole system speeds up. So this starts to develop some supracellular aspects to it, maybe, but something is going on at the collective level, and that results in no boundary outgrowth here, high boundary outgrowth here. So what we're starting to see is that the level of collectivity or the type of collectivity can make it easier or harder for us to work with the system, and we're trying to find ways to actually fix that now. So the idea would be to say, find something that's causing this and try to target it, inhibit it, and rescue nice, clean, forward electrotactic motion. So we hypothesized it was cell-cell adhesion because that seems plausible for these types of collective behaviors, and we think it's competing with us. So we want to see if we can perturb it to create some sort of effect. So we take the keratinocyte system, and there's a nice feature of it, which is you can grow it in low calcium or high calcium media, which mimics sort of different levels of available calcium in the skin. And in low calcium media, it acts somewhat mesenchymal, and in high calcium, it acts much more epithelial. Cadherin in the junctions itself tracks with calcium in the media, so we think it's fair to say that we're improving junctional strength, although that's a little bit hand wavy. But okay, that's the system we work with. These are zoom-ins of the corners. You can see the differences in morphology, but otherwise cells are seeded and worked with the same way on a fibronectin substrate. Now, if you do what we've been doing, this is what you get with basal calcium. This is the more mesenchymal collective. This is what you get if you start to look at what happens in a more collective tissue. This is kind of the limiting case, the extreme version of what happened with MDCKs. So this is quite different and surprising. And what we wanted to do was to start understanding where that comes from, what it means, and can we fix it? Because we actually made it worse than the MDCK case. Now it's collapsing on itself. And so what we see is as you increase the calcium levels, your speed during electrotaxis gets lower and lower. But your order doesn't change. Your directionality, your sort of signaling response and polarity is fine, but the speed that you move at is slower. And these outgrowth chymographs from the leading edge versus the trailing edge here emphasize this. But we like these heat map chymographs a little bit more because they're kind of averaged through the tissues and they show you really what's happening across the population. So this is control data up here before the field turns on. Field turns on at the dotted line. And then in a low calcium system, you see this marching in formation where the trailing edge moves forward and the leading edge moves forward, and the speed is relatively uniform and high. At high calcium, which is the highest you'd normally culture these at, you see pronounced retraction at the leading edge, lower speeds overall, and a little bit of advance at the trailing edge. And maybe there's elements of supercellularity again then, because you're starting to set up domains of behavior. And at intermediate calcium, you have medium speeds, some trailing edge motion, still some retraction, but a little bit of recovery. This retraction is extremely dangerous as well. It's this catastrophic failure. You get blebbing. We see that the lamellipodia always snap back before their associated nucleus or cell body moves. So we think that it really is something that's targeting lamellipodia. It could be traction forces build up too heavily there. We'd love to do some mechanobiology here. And it's really not nice for the cells. I don't know if this movie will play, but if we do a live dead stain during electrotaxis, we see PowerPoint freezing, but I'm going to just manually move it to the edge here. The edge dies. So let's just step back. We are trying to move something around. There's a lot of fun biology we could do with what we just looked at. But if our goal was to move a tissue around and kind of demonstrate practical control, this is not a good thing. So we need a way to manipulate that. And so we ask, how can we engineer collective cell behaviors? And I told you at the beginning, it's great to think we can just synergize with what's there. And I like this, it's nice and peaceful when it works, but sometimes if you can't join it, you need to beat it instead. And now we're gonna say, well, what happens if we actually mess with this? So we think it's E-cadherin, so we provide an antibody blockade against E-cadherin. We use a DECMA treatment, which is a blocking antibody to interfere with junctional adhesion and hopefully weaken it. And what we saw was this is medium calcium, no retraction at all, high speeds, and it rescues the kind of mesenchymal or low calcium phenotype. So we do think it's E-cadherin that's playing the key role here. Again, order doesn't change, but speed goes up. Even the low calcium cells that have very weak adhesions, and when you look at them in fluorescence for E-cadherin, it's really difficult to find junctions. Even they go much faster when you block extracellular E-cadherin. So it's doing something that may be a combination of mechanical and signaling roles here. We were not able to get DECMA to work well with very strongly junctioned high calcium tissues, possibly because it can't get in between the cells. But so we said, what's something a little bit more transient and reversible than the antibody treatment? And that was a calcium chelator. So we said, well, what if we just transiently disrupt junctions with a pulse of an extracellular calcium chelator? Can we rescue function? So we go from this to this tissue here, which is treated with a pulse of BAPTA, and then the cells are grown during electrotaxis in low calcium media, and then we switch back to high later. So there's a lot going on there. we deliberately break the collective behavior that the tissue started with, move it when it has weaker cell-cell junctions and sort of a less coupled collective motion, and then reassemble it at the end by adding calcium, high calcium back. And there's a lot of weird stuff here too. I don't know why it contracts vertically like this. It still has a tiny bit of retraction, but it's such powerful motion that it just pushes itself forwards. And you've got a lot of trailing adhesion attachments at the end because it's moving faster than those remodel. But that's very powerful and that's extremely compelling. And this is a tissue moving almost millimeter distances just by weakening the collectivity. So the sort of summary at this point is that there are three areas here that we're trying to explore with this type of thing. And the first is technologically, what are these devices going to allow us to do? What are the limitations of the devices? If we had more electrodes, would they actually help us? So this is more of the biophysics types of questions here. Model systems. Electrotaxis is really still in its infancy in terms of what it's been tried with. We're really excited to try some 3D models. We've started building these 3D epithelial cysts and vascular networks, and we're trying to see what other collective behaviors can be biased by these stimulation cues. And someday, of course, it would be really nice to have an electric Band-Aid that might actually modulate wound healing. There are some approved medical devices around electricity and bandages, but the clinical results are extremely unclear. And it seems like we need more fundamental knowledge to better optimize how those devices work. So I'll stop here. I want to acknowledge my wonderful group that grew in the last 2 years a lot faster than I would have thought possible. This is our new Zoom lab photo, because that's our new lifestyle. A lot of the work you saw for electrotaxis was spearheaded by Dr. Tom Zeidel, who's a postdoc, and Gowan Shim, who's a second year or third year now grad student. Matt Heinrich worked on some of the collective motion at the beginning, along with Ricard Alert, and Irving Miramontes was working on some of the cytoskeletal imaging. And this is a water bear because we do have projects in water bears looking at their gait, and they are just collections of cells collectively migrating. Thank you very much.
Thank you, Daniel. That was super cool. Yeah, just a reminder to everyone, if you have any questions, just put it in the chat on YouTube or on Zoom and we can relay them across, or you can write questions if you ask it yourself. Maybe just a quick question from me. So in this collective behavior where you have got the high cell-cell adhesion, do you see these front cells that everyone's crashing into to be any different when you change the strength of your electrical signal?
That's a fantastic question. We've started to try to get preliminary data for that. What we have seen, so I don't have a clean answer. We're going to look and see if retraction speed or something changes. But what we have seen in terms of speed is my screen still sharing, or did PowerPoint?
No, we lost it.
Okay. There's a lot of strange data on just general electrotaxis and field strength. And I don't know if PowerPoint's going to let me open it again, but if you increase the field strength, the overall speed goes up. And for MDCKs, it's relatively linear. We haven't actually found the maximum field strength. We've gone from 0 to 5 so far. We can see up to a 400% increase in migration speed. But tracking down what's happening at the leading edge specifically has been a little difficult. So what we're excited about is the high mag imaging to kind of see something. Is there a marker you'd be particularly interested in there? Because we were going to start with focal adhesions and or like a F-actin, but
I was just wondering that. I mean, I was expecting. I'm imagining there's going to be a limit at which point the cells can't move any faster, which is probably, I guess, related to actin rates or focal adhesion turnover or something like that. Yeah,
it's really surprising to see something like an MDCK cell go from 20 microns an hour to 100. So something really weird is going on. We'd love to do some traction force, especially at the leading edge, to kind of see if there's a difference in contractility or something that leads to this, or if it's possible it's signaling related, because those cells have a rear cadherin adhesion and then front lamellipodium, where cells in the bulk don't. So we really don't know what the field is doing at the collective level there.
I'll go to one of the questions. Oh, do you want to go, Jan?
Oh, well, I was just gonna say, yeah, I totally agree that looking at tractions and stresses would be really, really cool in the system. So we have a question from Amanda Stephenson. She says, the overlay of the lamellipodia in the circle assay, can you elaborate on what you're seeing there in real time?
Sure. Thank you for humoring the large amount of data that I stuffed in. We just got very excited about it, and I was trying to share. I think you're talking about this one. So assuming that that's right. I don't know.
I think it was back when you had, it was there's sort of a colorful bunch of lines for the rear retraction. Oh, I see it. Slide 68. I think that's when the
oh, this thing. Oh,
yeah,
sorry.
I think that's when the question
Okay.
popped up.
This is very crude low mag phase imaging here, and we just tried to see a little bit of quantitative data from that. So you see a cell spinning in the middle. It has a very sharp phase contrast boundary at the trailing edge. So what we did was just what's called a temporal color code in Fiji. You just Z-stack with a different color for every plane, and that shows you the brightest structures essentially stacked on top of each other. So these bands here are where the trailing edge is at any point in time. I'm sorry that the color bar that was supposed to show 0 to X hours wasn't showing up, but that's what this spinning is. And it's just a way to visualize the movie there. And it looks a little bit to us like the lamellipodium almost curls backwards right at the beginning of this and starts to spin, but that's something else we really want to see at high magnification.
Bill Gia has asked if the collective response to the electric field depends on gap junction and connectivity.
Also a great question. I think that's probably very cell type dependent. Min Zhao's group at Davis has done really impressive signaling work, and they looked at inhibiting gap junctions in MDCKs at least and didn't find compelling data, I think, that they were responsible for this. But I imagine that in some cell types that might matter. Something to keep in mind is that, at least in the sort of classic models of electrotaxis, the membrane impedance, the resistance of the plasma membrane is so high that you're not really biasing the flow of ions directly into the cell. You're likely acting on receptors and moving them, but you're not passing a current inside the cell in a simple way, at least. So that might be why gap junctions aren't an obvious target.
And just to follow up on that, so you said that it's still an open question, right? Like what those mysterious receptors or molecules might be that are potentially moving.
Yes.
Or localization. Okay. Are there any candidates, or is it just totally unknown?
I don't think there's a consensus. I think it's very much cell type dependent. I think so. Alex Mogilner and Julie Theriault did a study maybe 7 years ago on just the kitchen sink of different inhibitors and modeling of what might be happening in single cells, and they identified something similar to a syndekin as meeting the profile that might be allow for electrophoresis, but it's hard to say, and the imaging is a little bit tricky. There are some studies that show a concanavalin A receptor. If you tag that with fluorescent con A, those will move across the cell membrane in a field, but that just means something's moving. It doesn't tell you what. And then I'm not going to speculate there.
Okay. Cool.
That's why we tried to go in reverse, and we said we know the cytoskeleton and focal adhesions have to do something, so we're going to try to start there and go backwards, because at least that sounds plausible.
Yeah, it makes sense. Sorry, that was me just jumping in. Vladislav asks, I guess this is for the MDCK experiments, to what extent are the cells maintaining their cell-cell contacts in the sheepdog assay?
That is an excellent question as well, and probably so I don't have the movie here. We've done ECAD hearing imaging in ECAD RFP MDCKs, and there's not even a change in intensity that was obvious, but we have to go actually quantify it. But if you watch this phase contrast movie, there isn't an obvious change in the brightness of the phase boundaries. So their junctions are staying stable. But something we were very curious about was maybe going in and doing FRAP on a junction to sort of look at turnover times, which might indicate changes in tension. But we don't think it's damaging anything other than the leading edge in the keratinocyte model. And we don't see apoptosis anywhere else or increased extrusion or anything.
Okay, Brian Camley. Hopefully that skips anyone. Brian Camley has asked, when you play with calcium levels to get the high adhesion, do you still see the cells elongate perpendicular to the field?
That is excellent. No, we don't. And it's really, they're crazy messy cells though. So to do it better, we might do a mosaic with just a couple labeled cells in a population to kind of better see, because they kind of sit on top of each other, but it doesn't look like they orient the same way. And they do have strong cell-cell adhesion. And it could be that that means that, I don't know, maybe stresses don't equilibrate quickly enough compared to the lower calcium case. But what really was extremely surprising to us was that low calcium cells with the antibody blockade against ECAD had such a strong boost in speed, even though you can't really see junctions when you go in and look for them. So I'm not really sure what the junctions are doing or what the role of cell shape and contact is.
I guess following up on that, C. Chang has two questions. First, what's the advantage for the cells to orient perpendicularly to the migration direction?
That is something we would like to know as well. And what we wondered, and maybe attraction force data would show that, or something in the cytoskeleton, we thought maybe it provided greater speed or stability, but we're not clear yet on the timing of where that orientation comes from or when it comes in. So if you watch this cell here, it really starts to elongate. You get a lot of ruffling at the lamellopodial edge. And I know Julie Theriault's done some work with keratinocytes and how the shape affects kind of motion stability, but I don't know if that's what's happening here. So if you have any ideas, let us know.
And the second question from C. Chang is just about the calcium. Do you see any effects of calcium without the electrical stimulation?
In terms of control collective migration, they'll move a little more slowly. Depending on how long they're in it, you can have changes in proliferation, but mostly the control cases just kind of sit there. So we don't see obvious changes to behavior other than the junctions increasing and the cells getting a little bit flatter, but and the cadherin levels going up. But I think that's why I like the antibody blockade, because there could be signaling differences, and I think we showed that at least ECAD by itself can account for the effects.
So I have a sort of a follow-up to that, because I'm trying to wrap my head around this. So when you have the keratinocytes, they're much more responsive to the they're able to be herded when they're more mesenchymal and they don't have their junctions, right? But for the MDCKs, do you see the opposite? I would guess you might see the opposite effect, where if you inhibited the junctions, you wouldn't see the response, or
So we haven't tried that in MDCKs.
Okay.
I think there's a recent study from Northeastern, and I think Min Zhao also tried this a long time ago to look at cadherins. And in MDCKs, they don't electrotax without cadherin. They also have strange effects where small numbers of MDCKs or individual MDCKs also don't electrotax well. And it could be that there's some sort of combined signaling benefit, or that their mechanics and tractions are so high that they don't reconfigure. I'm not sure. They also don't retract at the leading edge.
right?
And I don't know how you directly compare adhesion strength between them and the high calcium keratinocytes. So I'm not sure how to speculate.
Just to sort of organize my thoughts, it seems that the role of junctions is cell type dependent. Would you agree with that?
That seems, from what we've seen so far, yes.
It's really, really interesting. Thanks.
Yeah, so you really can't generalize with this. It's kind of, I mean, it makes sense, but the collective behaviors are so different that, like, high calcium keratinocytes don't show any of the long range correlation length that MDCKs are famous for. So you don't have that 200 micron correlated fluctuation domains inside the monolayer. They just kind of sit there and look bored. So it's, and they have strong junctions too. So it's just very different behaviors.
Cool. think it's the last question. If anyone has any others, just pop them in before we finish up. Yutaka is asking, in the wound healing assay, are you wounding with a scratch or are you removing the block? He's asking because physical damage greatly changes cell behavior.
Yes, that's great. We do barrier removal for those. So we deliberately try not to damage the matrix and not to damage the cells yet. I think if we go in and do a scratch, then we'd start to get more signaling effects. And so that's something that's definitely worth doing. But to keep things controlled in the first pass, all barrier removals.
So in the paper, I put a link. They have special technology to damage leading edge cells without affecting extracellular matrix. Maybe it's useful for you.
Oh, cool. That could be fun. I will take a look. Is it in the chat?
Yes, in the chat. Yeah.
Thank you.
No problem. Because it's very surprising difference what the damage causes, and this paper have a nice control.
We have one method that we've tried, which is to, if you grow MDCKs inside a stencil for a long time and you get a nice monolayer, and you pull the stencil off, you have about 1 hour to 3 hours before lamellipodia form. And if you do any electrotaxis or migration assays during that time, you have no restoring force, or you have weak restoring forces at the leading edge. And we actually see sort of retraction in those cases. So it could definitely be that stuff at the leading edge is affecting this. One thing I also didn't necessarily trust in my original study was we did it on tissue culture plastic. So there would be time for serum to deposit on the dish as well. I wasn't really happy with that. The keratinocytes are done entirely on fibronectin matrix, and we know the matrix isn't damaged. So whatever's stopping them from moving outwards, it's not that they can't attach to the substrate well, because the substrate's definitely there. But I'll take a look at the paper. Thank you.
Yeah, thank you. And maybe you can add some reactive oxygen species.
That could be fun to kind of change the electrochemistry. Yeah, if you
Yeah,
want to talk
always
afterwards.
After wounding, always it happens.
That's a good point. That might be very fun in a fully junction system too.
Yeah.
Okay. Thank you.
Thank you.
Can I ask a quick question? Does galvanotaxis work with adhesion-independent migration? Like if you're in a cell that was confined, would it work?
I think there are examples of that. I know it works well in Dicty, and I'm embarrassed, but I don't actually remember how Dicty forms attachments to the substrate. Is it of a blebbing mode, or is it focal adhesions?
It's a podosome. I think it's like there's a protrusion generated.
So it definitely works with Dicty, which would be a different form of locomotion. I'm not sure about general amoeboid motion, but it's been shown in a number of species and examples. So the answer is probably yes. C. elegans responds to it as an organism, and you can sort of adjust their turning, but that may be a different process.
Cool. Jen, do you want to end the YouTube?
Yeah, well, thank you everyone who's still here. We'll see you guys next week.